Hey everybody, it's Mrs. Mancuso, Merm here, ready to do a little lesson on DNA. So let's say this is you, all happy because you're in AP Bio, and here you are with curly red hair. So how did you come to have this curly red hair? So this is one of your phenotypes. It's one of your traits, and although it isn't due to just one gene, let's say it is. So let's say it's due to having this genotype, which means that out of all of your DNA, the one gene that is coding for red hair in this case is due to a dominant allele. So the big A is the dominant allele, and you have two of each type of chromosome. So they come in pairs, and let's say the hair gene is in this location right here. And if that sequence is the same on both of them and it codes for red hair, you are homozygous dominant for red hair. So today's video is all about DNA, and we're going to be doing genetics and all this interesting stuff about genotypes and phenotypes. So let's get right into it. So the structure of DNA. We talked a little bit about the scientists that helped us come to understand the structure of it. And we're going to spend more time today going into details of the structure of it. So first off, some basic info. So where do you find DNA? In a eukaryotic cell like our cells, you find it in the nucleus, mitochondria, and chloroplast. But in a bacterial cell, if this is bacteria, they don't have a nucleus. So in a prokaryotic cell, DNA is usually circular. So this is my double helix. It just goes around in a circle. And then there's usually some extra DNA, which is also circular, called plasmids. And those are the ones that are really likely to enter and leave a cell. So bacterial cells can actually pick up other genes from their environment really easily. All right, makes up chromosomes. So we were just starting to talk about this in my slide over here. So we know in humans that we have 46 chromosomes. They are really pairs. So there are 23 pairs. And those chromosomes really just look like a strand. In reality, it is a double helix. So it does go around like this. At one point, there is a centromere. We call everything in the top half of it the P region, and everything under that the Q region. So they've actually mapped out the human genome, and they can tell you the location of different traits based on the chromosome number. So we could say like, hey, this is chromosome 5, and it's found in P 111. So that's the location, that spot right there. And these chromosomes are all made up of DNA, which means they're made up of base pairs. So that DNA has to fit into a nucleus. So in order to fit, it's arranged into what we call nucleosomes, and that's how it fits in such a small space. Because if you stretched out DNA, it would be really long. So it's like clusters of DNA wrapped around histone proteins. So if this is my histone protein, the DNA would coil around it like that. Sort of like picture a spool with thread wrapped around it. And so if you wrap it around, you can fit it in a small space. Now, there's two kinds of words that we use for chromatin. Chromatin is our DNA. So you could call it heterochromatin or euchromatin. I think of the H as meaning hidden. So heterochromatin is DNA that's really tightly coiled, and it's not very likely to get expressed because it's wrapped around this nucleosome super tight. So transcription factors and other things like enzymes like RNA polymerase can't get into that space. And then euchromatin is another term to describe DNA, but this one is expressed. So H for hidden, E for expressed. So euchromatin is loose around it and much more likely for enzymes to get in that space. But then sometimes DNA is methylated. And if it's methylated, think about it like a little tag that's coming off of it. A methyl tag is a CH3. So here's the tag attaching to the DNA. And if it has these 3 H's around it, it's very nonpolar, which means it's going to hide that section of DNA so it won't get transcribed. And so you might think right off the bat, like, oh my gosh, why would we methylate a gene? And it's so that we don't express it in that area. So for example, you have DNA that's in your eyes, or sorry, that has to be expressed in your eyes, but that same DNA is also present in your stomach, and that section of DNA doesn't need to get expressed because you do not need the same genes to be expressed in your stomach that you need it expressed in your eyes. So you can inactivate them. All right, so now we're going to get into the actual structure of DNA, and I figured we would just go ahead and start drawing. I think drawing it is one of the best ways to learn the structure. So we start off with, we know that DNA is made up of nucleotides, and nucleotides have 3 parts: phosphate group, 5-carbon sugar, and a base. So let's do a phosphate group, which is PO4. One of the O's is a double bond. So it looks like that. So that's my phosphate group. That's attached. This will be my 5th carbon, which is in the branch. And then here's my 5-carbon sugar. So 1, 2, 3, 4, 5. You learned how to number the carbons in a sugar before you go away from the branch on the oxygen. So find the oxygen. The side away from the branch will be carbon 1, 2, 3, 4, and 5. So that's deoxyribose. And then I need a base. So one of the bases looks like this. So do an N, and then 5 sides like that. This is going to be another N, and a little line there, and another N right here. No, I don't want to make that an N. Sorry. I actually want to make the other. I'm going to make this adenine. So let's see here. Boom, boom, boom. 6 sides on this one. All right, we're going to do a double bond there, an N here, and another double bond, an N here, another double bond, and an NH2 here. Okay, that's actually adenine's structure. And then, so that's one nucleotide that we just drew. Now we have to draw another nucleotide on this side. So this time we're going to start with the phosphate group down here. And that's attached to the 5-carbon. So then this will be my O. Oops, I made it have too many sides. All right. Should have had a little picture here next to me. Okay. Carbon 1, 2, 3, 4, and 5. Okay, that looks better. So just so you know, the other phosphate group will attach here, and the other one will attach there. All right, the one is always attached to my nitrogenous base. So this time it has to be thymine, and thymine looks like this. So it's got 6 sides like that. There's a double bonded O here, an NH here, another double bonded O there, a double bond there, and a CH3 there. Okay, and then there would be hydrogen bond there and there. So 2 hydrogen bonds. Okay, so what we have drawn is 2 nucleotides. I don't know why I'm getting this box around it, but oh well. All right, so here's one nucleotide. This is the 5-carbon sugar. This is the phosphate group. This is an adenine. And then on this side, this is my phosphate group. This is my 5-carbon sugar, and that's my thymine. The sides are anti-parallel, which means they run opposite each other. On this side, the 5 is above the 3. So this is my 5' to 3' side. And on this side, you'll notice the 3 is above the 5. So this is my 3' to 5' side. And so one side looks upside down compared to the other. The outside, the backbone is always the sugar phosphates, and the middle is always the nitrogenous bases, and they're held together with hydrogen bonds. So I know you want to add all that to your notes. So I organized this all. I know you might have to slow me down, but you can always just pause to add this to your notes. So DNA structure, it's a nucleic acid, which means it's a polymer of nucleotides. Every nucleotide has those three components we were just talking about: a phosphate group, a 5-carbon sugar, and a nitrogenous base. And that's why DNA is negative too, because those phosphate groups with all those oxygens on them tend to be very negative. All right, and then the nitrogenous base, I got to do a little bit more details on. So that's back when we did biochemistry, we talked about it as a ring of carbon and nitrogen. And the double rings are called purines. They are adenine and guanine. And then the single rings are the pyrimidines. Those are the thymine, cytosine, and uracil. T's, C's, and U's. So the structure that DNA takes on is a double helix. It has 2 strands like we just drew. They run anti-parallel to each other, makes one side look upside down. The strands are labeled 5 to 3 and 3 to 5 based on the position of the sugar. You always number the carbons in the sugar with the 1' attached to the base, and the counting always goes away from the branch. The branch is always the 5'. The 2 sides of the DNA, the 2 strands, are held together by hydrogen bonds. The hydrogen bonds are between the bases. There's 2 hydrogen bonds between A and T, and 3 between C and G. I think an easy way to remember that is C is the 3rd letter of the alphabet, and C forms 3 hydrogen bonds. I've seen this question very often asking about separating strands. So like if you imagine that 80% of a strand of DNA is adenine and thymine, so 40% adenine, 40% thymine, that'll be a lot of 2 hydrogen bonds instead of the 3 hydrogen bonds. There would only be 20% C's and G's, so 10% of each. And so there would be a lot less of the 3 hydrogen bonds. So it would actually be easier to separate if you had that. It would take a lower temperature to separate it apart. The 3' to 5' strand of DNA is used as the template for transcription. So transcription will be a whole nother video that we do showing how you make RNA. But you always want your RNA, because that's only single-stranded, to be 5 to 3. All right, and then next, DNA is our code. So you probably recognize this chart down here. This chart is always to be used with our mRNA codons. So as we do transcription, we will make messenger RNA, and then as we do translation, we will use this box and figure out the order of amino acids in the protein. So DNA is really, I mean, like the entire purpose is it is the directions for building a protein. And the primary structure of a protein, you remember, is the order of the amino acids. So if I just give an example, like if my DNA, my template strand is going to be my 3' side. So if this went A, TCG. All right. That means my messenger RNA, it would be 5' and it would go U, because there's no T's in RNA. And then it would go A, G, C. And so to look that up, I look up U, the first letter U, the second letter U, and the 3rd letter U. So U, it would be PHE. That would be the first amino acid. And then AGC, serine. So serine would be the 2nd amino acid. And you would just keep going and going and going until you hit one of these 3 stop codons. All right, some more facts about DNA. First, you have to copy over all your DNA if you're going to make a copy of the cell. So if your cell is about to divide, if it's about to go through mitosis or meiosis, every daughter cell is going to need all the protein making directions. So you got to copy it. That's why S of interphase exists. And DNA is the same for every cell on Earth. It doesn't matter if it's a prokaryotic cell from a bacteria or if it's a cell in you. Everybody has the same DNA. The only difference is the order of the A's, T's, C's, and G's. So it shows a common ancestry. It also allows genetic recombination. So you can like take a gene from a bacteria and put it into another organism. And that's going to come up later in this unit. And it's been a huge part of agriculture and cancer studies and all sorts of things. Another thing I just thought was interesting is the Human Genome Project. They figured out the entire DNA sequence. They started in 1990 and they finished in 2003. So they now know all the genes that make up the human chromosomes. And it's also cool to know that you have error checking and proofreading enzymes. So there's so many levels of regulation to make sure that there's not mistakes, and when there are mistakes, to fix it. And then I just put these on here because I just wanted to go over a diagram that is from a book instead of one that I drew. So first of all, they're calling this the 5' end because my carbons, 1, 2, 3, sorry, my 2 didn't come out, 4, 5 is the branch. So the 5 is above the 3. So this is 5 to 3 in this direction, which means on this side, 1, 2, 3, 4, 5, the 3 is above the 5. So this side goes 3' to 5'. These yellow circles, this is our phosphate group. The orange is the deoxyribose, so that's the 5-carbon sugar. These dotted lines are the hydrogen bonds. This is a purine because it's a double ring, and this is a single ring, so this is a pyrimidine. And this would be one nucleotide. If I have one nitrogenous base, one sugar, one phosphate, that makes up one nucleotide. And so DNA is a double strand because you have 2 strands of nucleotides. And I encourage you on the day that you are working on DNA structure to just draw DNA like this. I think it's great practice, and it gets you used to the purines, the pyrimidines, how the bonds fit together, where the attachment points are. It helps you really visualize 5 to 3 and 3 to 5. All right, that's it. Enjoy your day, guys. Bye-bye.